Hi guys, so this is going to be chapter 20. This is the plant anatomy and growth lecture. Now with plant tissues, you're going to notice that there's quite a bit of differentiation in plant tissues. They're not all going to be doing the same thing, and they're not as simple as we've made them seem in previous units. So if you're thinking back to Biology 1 from high school bio, if you're thinking back to even semester 1 of 1408, the plant cell is pretty complicated. It's got this special tissue called a meristem. Okay, that meristem tissue is able to divide and actually be with the plant the entire life, but its ability to differentiate is its big claim to fame. So for humans, we tend to have stem cells that those, whether they're in the bone marrow or whether they're embryonic, they spend their lives specializing. When it comes to plants, you can have apical meristems, okay, or lateral meristems, but they end up differentiating into many different tissues. So the apical meristem, for example, can become your epidermal tissue. That's the outer covering of the skin of the plant, so to speak. They can become ground tissue, which is basically the body tissue, or the transport tissue, which is the vascular tissue. And so with the plant, with the epidermis, you're going to see that it's pretty much covering the entire body of the plant. Whether you're talking about leaves or roots or stem, they're all covered by that way, but we recognize them the most around leaves. That's when they have a waxy cuticle. So the main point of this is so that you don't have any water loss, and it's also helpful in preventing disease. A lot of viruses and bacteria are not able to penetrate into the actual cell. They'll just end up infecting the cuticle, and that's one of the things that happens there. And again, for water loss, you're going to see plenty of times through this PowerPoint, and we talked about it in the fall, that the plant itself is going to have stomata that's going to release gas and also prevent water loss as a way of not letting that water out. So these are different types of tissue that you're going to see. We've got root tissues, we've got guard cells, we've got these trichomes that are the stoma of the leaf. And then once you get to different types of plants that are undergoing secondary growth, eventually you're going to see some cork or bark that make the thicker trees, and the stems become trunks. So as you can see in this one, you've got chloroplasts in the guard cells, and basically these cells are located on the bottom of the leaf for gas exchange. The plant needs to be able to get in CO2 and release O2, but these cells are able to block water from being lost or allowing water to get out in the case of there being enough water and the reactions have already happened. You're going to see later on that quite a bit of the water that is absorbed by the roots of a plant gets lost, even though they have these guard cells, but that's one of their ways of not allowing themselves to lose too much. And this one right here, this shows the cork of an older stem, which eventually is going to become a bark of a tree. So as this stem starts to undergo primary growth, which is the actual growing of the tissue, then eventually it's going to undergo secondary growth, where it's going to replace those outer layers with cork, and then eventually it's going to get rougher. And so that suberin is what is allowing it to be chemically inert. And so this is not what we would say, the plant is not dead at this point, but that's not exactly live active tissue. Think of it in terms of hair or nails. That on a human being or a mammal, hair and nails, those tissues are not really active. They don't have any nerve tissue and things like that. That's the same thing with the plant, except there's no worry about nerves. Instead, this is not a photosynthesizing part. It's not really a transport part until you get to the inner portions of the cork. So when you're talking about right here, this portion is going to actually do a lot of the transport, but out here, the actual cork and the bark, that's not going to be doing much. And so when we say ground tissue, I mentioned that in the first slide, basically that becomes the bulk of the leaves, the stems, and the roots. So there are three types. It's very important to know the different chymas. We have parenchyma, collenchyma, and sclerenchyma. So we'll go through those. The parenchyma tend to be your least specialized. I like to think of them as kind of like the body cells or the fat cells. We're going to learn later when you go through different tissues. It looks a lot like adipose tissue. It looks a lot like the fat tissue in many animal organisms. So that's what this ends up doing, the function for plants as well. It's got a lot of chloroplasts. They are photosynthesizing, so they're not dead cells, but it tends to be kind of these colorless cells that are just hanging around. So these make up the body, or if you don't want to call it the fat, maybe more like just the muscle type tissue of the plant. So that's what we mean by ground tissue, and the parenchyma is one of those. So that's one. Then you have the collenchyma. These are the ones that are more flexible. These end up supporting, and it's important to understand this one, the immature regions of the plant. So as the plant is growing, the collenchyma is what gives it that flexibility. Okay, that's different from the parenchyma, because the parenchyma is just the main body and it's least specialized. But then when you have the sclerenchyma is the one that's going to be non-living, mainly lignin, and it's going to support the mature regions of the plant. So when we're talking about the bark, the woody stems, that's what we're talking about, the sclerenchyma. So it is important to note the difference between those three. Now, we've talked a lot about vascular tissue in the last chapter, and in the lab we talked about xylem and phloem. Okay, so we know what these are. Xylem ends up transporting your water and minerals, but it's also important to note the direction we're going. So here we're going roots to leaves. That is a big deal. Then there's two different types of the xylem. You've got the vessels, which are these large pipelines. Okay, so you're going to see that in the next slide. They're very big and spacious that allow for water to move in. And then we also have tracheids, which are smaller, thinner pipelines. They basically just kind of allow water to move back and forth and allow for pressure in the stem cells. So it's important to note the differences here. And I like to think of these, in the next slide we'll look at them. I like to think of these kind of like blood vessels. When you have arteries, those tend to be the really big, spacious blood vessels that allow lots of blood to be passed through and around the body. But then, besides having arteries, you also have capillaries. Those tend to be the smaller ones. That's what the comparison would be. So these larger vessel tissues, they are allowing the water to be pumped pretty quickly through these big tubes and these vessel elements. Whereas the tracheids, they're allowing the water to kind of fill in to the rest of these through these pits on the sides right here. So it's still moving, but it's just kind of like filling up the pressure through this little pipeline here, and then it's moving up to the rest of the plant. Now, as we remember phloem, the important thing to understand here, the direction goes from leaves to roots for the most part. Now, this does change when the plant is an embryo. So if we're talking about a bean, the phloem does allow for the sugar in these thicker root cells to move up before it's able to photosynthesize. But once the plant is grown, it's going to be from the leaves down to the roots. So they have different cells as well. They don't have the same tubes as xylem. They have similar ones, but they do different jobs. So here we have the sieve tubes. These are these big pipelines with no nuclei whatsoever, but they're always attached to a companion cell. These are the ones that have the transport function. They're the ones that have the genetic information. So you can see that in the next slide. Here we've got the sieve tube, and we'll talk more about that in detail later, but this is basically a big giant pipeline and what we mean by sieve is kind of like a filter tube. So it's able to allow that movement down of sugar, and then these companion cells have the nuclei that are able to have the genetic information that produce these two together. And every time you have a sieve plate, it ends up kind of filtering through and letting that pass through. Now, we think of plants as pretty simple things, but they technically have organs just like we do. Like you think of an organ, you probably are thinking of your heart, liver, lungs, intestine, stomach. Those are the organs that we're familiar with, but plants have two different systems, the roots and shoots. Those are important words to know. Roots are basically everything underground. The shoot system is everything above ground. That's your stem, your leaves, your flowers, and eventually, once it's fertilized, your fruit. Okay? Every plant undergoes primary growth. That's a big deal to understand. Okay? Primary growth is that plant basically going from your seed into your stem and then forming your leaves and flowers if it's a flowering plant, or your gymnosperms. They could have just their pine cones. So those are primary growth. It's basically the tip of the root causing it to grow in length, and at the terminal bud, so basically they're growing at two ends. They start off as a seed, one end is growing in the root system and one is growing in the shoot system. So you can see that in the next one. And here it is. So there's the terminal bud. Okay, that's the one that allows the plant to grow tall. And then the lateral bud, which allows plants to grow sideways. So they're able to grow their leaf and their petioles off to the side. And then down here, you have the lateral roots, which are growing sideways. And then the root tip down there, that's the primary root that grows down. So some plants have varied their root systems. You have a thing called taproot where they basically just go straight down. But basically, to finish it all off, the root system spreads out. It's able to have all that nutrient collecting and water collecting, and that's what you need those root hairs for. Excuse me. They allow for more surface area for collection of the things you need to get up to the rest of the plant. So there's kind of the microscopic view as this starts to happen. So this is your lateral bud. Those are the ones that are letting the plants grow off to the side. So that's what this thing is right here. Okay. And then you've got the apical meristem. That's the one that's at the top, allowing the plant to grow out. And these immature leaves that eventually will pop out and become your plant. This looks like it's a dicot because it's got the two embryonic leaves. And that's the next thing. So one word you're going to have to be familiar with is a cotyledon. Okay, so the cotyledon is basically your embryonic leaves. Those are, when you're a seed, that seed is going to end up germinating. And when it starts to germinate, is it going to germinate one leaf to start? That's a monocotyledon. Or is it going to germinate two seeds to start, or two leaves to start? That is a dicotyledon. You'll also see it labeled in many of these as a eudicot. Same thing. But anything with the word dicot is two cotyledons. So you'll see a lot of comparisons in the next few slides. If you look here, the monocot, or the monocotyledons, they have one of these embryonic seeds. So this tends to be like a grass seed or a rice seed, sometimes wheat and corn, but they're very, very distinct because when they start to grow, they will have one leaf coming out. Whereas if you remember last semester, we grew a lot of bean plants with our Biol bottle project. Those ended up having two leaves when the bean split open, whether it was lima or pinto or any other type of bean. And those two leaves were the two cotyledons, okay, in the seed. Those tend to be your dandelions, your oak trees, and several other different types of flowering plants, okay? And so there's a lot of different characteristics you're going to see here. So in the roots of the monocots, they are very distinct, and we're going to talk about this in detail, but they have a nice ring of xylem and phloem. So it's a very organized root. Whereas the roots of a eudicot or a dicot, they tend to have this little star-shaped ring. So this ring for them is not really a ring; it's their phloem, it's the green part stuck in between the arms of the xylem. Okay, so their roots do look different when we look at a cross-section of a slice of these things, and then the stems as well. The stems have vascular bundles both. But those vascular bundles in the monocot, they're spread out all the way through. And then the vascular bundles in the dicot, they tend to be around the center in the middle of the plant. And you're going to see why in just a bit. We'll show you what that looks like. And then one of the things you're going to notice is the leaves have veins. The monocot veins tend to be a little bit kind of like the veins right here of the stem. They're parallel to each other. Whereas the veins of a eudicot, they're the ones that are more spread out from one central point. And the other big thing that's easy to tell what they are is by their flowers. If it's a monocot, there's going to be in multiples of 3, usually. Okay. If it's a eudicot, it's going to be in the multiple of 4 or 5. Okay? That tends to be the general rule. That doesn't always work, just depends on the plant and how many flowers it gets and its method of producing flowers. But that tends to be the general strategy. So the next big part of the organ system for a plant is going to be the 3 main shoot parts. That's going to be the leaves and the stems, and then that's two, and then the main part of the root system is just what the roots do. So the leaves they're the photosynthetic organism, because they're the green things typically, right? There are some plants that don't do that, but in general, the leaves are the ones that are doing the photosynthesizing. They tend to create these extra spaces. That's why you have these broader leaves that do quite a bit of collection of energy. And that's why the most evolved plants, like angiosperms, tend to have wider, bigger leaves, whereas the less evolved plants, like the gymnosperms and the ferns, they don't have as big leaves as some of the others. And that's the whole purpose of that, is to get some more of that CO2 out of the air. Okay? And they do receive water from their roots. And then we have this other type of classification amongst them called deciduous plants. Those are the ones that lose their leaves during the seasons. So when you've got here in the valley, you see a lot of that in the summer. Plants end up having to try to save their water, so they drop their leaves. That also happens in the winter when there's, you'd think, okay, well, there's a lot of snow in the winter, that's a lot of water available to the plant, but it's really not, because it stays cold and the water never melts. So it's almost like a drought for them, so they have to get rid of it. Whereas like a gymnosperm, like a pine tree, or any type of fir tree, like the ones you get for a Christmas tree, they're evergreens. They stay green all the time because their leaves are smaller and thinner, and they're not as susceptible to that lack of cold, because when you got a lot of surface area, that's great, but when it's cold, you're going to end up losing a lot of that tissue. So having those little needle-likes, those help quite a bit. Okay, and so for the leaves, you've got two main things to know, right? The blade, which is the wider portion that's going to be doing the photosynthesizing, and the petiole, which is the stalk that attaches that blade to the stem. So the petiole pretty much stays the same as is. The blade can get pretty complicated. So here it is in the next slide. As you can see, this is your typical blade. This would come from like a cottonwood tree. These have, this would be a dicot, because it's got that veiny pattern from a centralized location, so it's not parallel to each other. They do meet at a center, but this blade is the main leaf, and then you've got the lateral bud where the rest of the plant was growing from, and then this is the petiole. That's the stalk, or the part of the leaf that connects it to the stem. And the stem itself can be woody or herbaceous. That doesn't really matter much when it comes to leaves. But so this is where it gets interesting. So this is considered, this big section right here is considered to be the blade, even though it's got 5 parts. And this blade is divided into 5 leaflets. So this is what we call a compound leaf. Okay, a compound leaf basically just has offshoots from the petiole, and that can be various numbers. So depending on the plant, you can have a different number of blades, of leaflets coming off to make the entire blade. But again, they still have the lateral bud, still has the petiole, no difference there. Then when you have this one over here, now we've got a twice compound leaf. So here's what's happened. The petiole is right there, but then off of the petiole you have other smaller versions of the petiole before you finally get to the leaflets. Okay, that's these things down here. And then the whole thing makes up the blade. Okay, so this is a twice compound leaf. Here in the valley, we have quite a bit of those. Right off the top of my head, the easiest example is the mesquite tree. Okay, if you've ever gotten close to mesquite leaves, they have that compound leaf, and I believe it is a twice compound as well. So now, when we're talking about a eudicot, they tend to have characteristics depending on where they are. So if it's a temperate zone, that's an area where it does have some sort of a winter of some sort, probably a little more significant than our winter. But that has a waxy cuticle, which, like I mentioned at the beginning of the PowerPoint, its main goal is to prevent for water loss. And again, it's got the stomata, and it's wise to have the stomata on the bottom, because if you have the stomata on the top, that's where the sunlight is going to hit that blade more, and you don't want to lose water in the middle of a sunny afternoon. So it's important to keep that epidermis, first of all, for water loss, and then it prevents, and then also having the stomata under there prevent even more water loss. And we talked about this during lab, but the actual leaf is pretty complicated. So we're used to those big cuboidal plant cells, but that's not the case. A lot of the time those cuboidal plant cells can have different variations. They're not usually cuboidal, they're kind of globular, right? And so there's two different parts of what's called the mesophyll. That's the middle of the leaf. The palisade has those plant cells, but they become elongated. That's usually at the top of the plant, of the leaf. And then on the bottom, you have the spongy area, right around where the stomata is, or the stoma. That allows for the gas to come in and photosynthesis to really get its job done in the leaf. So you'll see that in the diagram here. And there's the structure, very quick and easy to understand. So you've got these bundle sheath cells in the middle of the veins, okay? But beyond that, you've got the stoma sitting on the bottom underside of the leaf, and every part has that epidermis, that outer layer. But inside, you've got these elongated cells that take up pretty much that top space, kind of insulating it from sun, so to speak. But then there's a bunch of gaps in the middle. So we saw this in the models in the class, in lab, but there's quite a bit of those space gaps, and I really like this slide over here. We'll get to that in just a second so you can see what it looks like in real life. And here is what we're talking about. So you've got that epidermis on the top, okay? You've got the lower epidermis on the bottom, and the stoma right here. Those are the guard cells that are able to let gas in and out. Then you've got that spongy mesophyll. That's a bunch of gaps in space. Like, see this right here? That is your airspace where it's able for that gas exchange to either come in or to leave. And then you've got so that's the spongy mesophyll, and the palisade mesophyll. That's the one that's more compact. It's got no gaps in between. It's still photosynthesizing, but it needs to get the gas from the spongy mesophyll for it to work. If it doesn't have that spongy mesophyll, it really won't be doing much. And so you're used to, like I mentioned, the cuboidal plant cell. Like, that's the typical cell we always have. The nuclei, the big central vacuole. You can always see the mitochondria and the chloroplasts, and then everything else, right? Rough ER, smooth ER, ribosomes, Golgi. But the thing is, that's not how they all look. As I mentioned, some of them can be elongated. They can be these big globs. So it's not something that is by far perfect every time. They can change and look different. And that's the close-up look of the stomata, or the stoma. And so these guard cells, they have their space right here where they kind of open up. And we'll talk about how that pressure works in just a bit. But through a change in pressure, the guard cell can either be opened or closed, and it will allow for that oxygen and water to come out while the CO2 is coming in. And that's something that all plants need to be able to do. So striking that balance between when is the stoma going to be open, when is it going to be closed, is an evolutionary tactic that the plants have had to adapt over time, depending on their environment. So leaves have many different purposes. In a viney leaf, you're going to see that the vines are not always the stem. Those tendrils tend to be the leaves. So the plant sacrifices its photosynthetic ability there. I mean, everything's still green, but it's using the leaf tendrils to kind of attach itself to either the fence or the host plant that it is infected. Okay. Then you've got the Venus flytrap. That's the one that captures little flies in there. So it has little triggers on each part, and then once the triggers are, I think it's 3 of them. Once they've been actually triggered, then the plant will shut itself, and it doesn't like chew the fly, right? It ends up kind of digesting it, excreting these digestive fluids. But that's all done by the leaf because the plant itself cannot gather nitrogen from the soil like it's supposed to. And then others will have no leaves at all. Their leaves are right here on the cacti. They're just thorns, and that's meant for protection. So in the stem, what we're talking about here, stems are still photosynthetic. Especially if they're green, they're going to be doing the photosynthesis, but they're not usually the primary form of photosynthesis. The primary ones tend to be your leaves. Now these non-woody ones, we call them herbaceous. They're the ones that only undergo primary growth. Okay, if it's going to become a stem like with bark and wood, then it's going to have to undergo secondary growth, and that increases the girth, which is a fancy term for how wide that stem is going to be. And so this doesn't always have to be the stem. It can also be the branches or the roots themselves that are undergoing secondary growth. So if you've ever seen a tree with roots that are kind of on top of the ground and they're really hard and made of wood, they've undergone that secondary growth too. So it's not limited to just stems all the time. So here we have some more stems. I mentioned this in a couple slides ago, but the cacti doesn't use its leaves for photosynthesis. It becomes thorns to make sure that nothing eats it, and the actual blade of the cacti, they are, they're the ones that are the stem itself that's photosynthesizing. So that's a weird one. And then the other one, potatoes, if you've ever had a potato, that actually is part of the stem as well. It's buried underground, and a lot of people for a long time thought it was a root, but it's actually part of the stem that's storing its food for the plant. And so when we talk about non-woody stems, those are, I mentioned, herbaceous plants. They tend to have a epidermis, but then in eudicots they have a weird thing. They have a cortex, and then they have a pith. So the cortex is this band of parenchyma. If you remember the first slide or two on these, the parenchyma is the body tissue of the plant. So it's not very specialized. It's just forming the tissue that is the plant. And then the pith is the actual middle portion. In dicots, it tends to be pretty well organized and clear, but in monocots, it's not always well organized, and so that tends to be one of the weak spots for a lot of people who are not able to see that. So that monocot doesn't really have it. It's just there, but it's not as organized as in the dicot. And again, what is the point of having vascular tissue? Those are the xylem and the phloem. The main idea is that they're vessels and tracheids that are supporting the system in the shoots. And our xylem and phloem, not only are they supporting, but they're moving things back and forth. In the eudicot, you've got a ring kind of in the center of it to show very clearly where things are going. And that's why eudicots tend to have these really woody stems later on, because they have the living material inside, and that's what's able to do the transport. Woody stems later on, you're going to see they're made out of bark, and so even though that outside is not so much living photosynthesizing material, it's still doing the work because of that center ring. Whereas in the monocot, they don't have that. Monocots are not really woody, so if you've got a large tall grass plant like a banana plant, which is basically just grass that's modified, its xylem and phloem is pretty scattered out in these vascular bundles, which means it's not really able to ever become woody. So it may become harder and more organized, but it's never really a wood stem. And so here's the differences, right? So these are the microscopic ones. In the eudicot, you see the pith in the center. So it's that right there. Now over here in the monocot, it's got pith, but it's not really localized. It's all in between. So that's what prevents it from later on becoming a woody plant. And then you've got the epidermis, the ground tissue, right, which is your parenchyma cells. Okay. But then if you look at the vascular bundles, the eudicot, they're on the outside. The vascular bundles on the monocot, they're pretty much everywhere. So depending on your type, that's going to really determine your, I guess, purpose and your methods of forming these stems. So these are all herbaceous ones. Then you've got the woody stems, as I mentioned. You've got to have primary growth, but then you got to have secondary growth to get wider. Okay, and that allows for those not just stems, but the branches and the roots to get wider. And just depending on when and where you got this vascular cambium, whenever that is activated in plants, that's when you've got the traditional wood stem or bark that you think of in a tree trunk. Okay, and so it forms these rings, and it produces more xylem and more phloem every year. Okay, and that's what allows it to pretty much stay growing. It stays parallel from the plant surface. And then of course, you've got the bark, which is that middle portion, the wood, or the outer portion, the wood is the middle, and then that pith is the very center. Okay, and that vascular cambium, that's the one that gets between the bark and the wood that allows it for more separation. So there's kind of your sectional diagram there, but in the center of it, you've got your pith, right? That's the body tissue. Okay. A lot of the time, you've got these two different types of wood, spring and summer wood, so you can actually tell the age of a plant technically with their trunk, but it's not always the age. You can really just see how many seasons it went through and whether or not those seasons were successful. Because more water, more CO2 available, that really identifies whether or not something is going to work out. And so we're talking about bark. Bark is that outer portion. It is semi-flexible still, so it's made out of this material called cork. And there is some phloem out there for the nutrients, and they have this chemical called suberin that allows it to stay semi-flexible. So if you've ever seen a cork board before, that's made because out of the same material, it's spongy and it's semi-flexible. So if a piece of bark, if you've ever seen that fall off a tree trunk, those pieces of bark tend to still be able to move a little bit. Okay, whereas the wood inside, that's the stuff that every year there's a growing season. That's what gets either wider or shows a wide ring or a thinner ring into these annual rings. So again, you can tell generally how a plant's done, and you can estimate, but there's no quick conversion to say, oh, it's got 9 rings, this thing must be 9 years old. No, it may have had 9 seasons depending on how much water was available, how long the plant has been there and stuff like that. So you can kind of identify, but it's not a straight conversion. Okay, and for roots, one of the things you're going to notice is you can't have a big tree unless it's got a long and big root system. So depending on the environment, obviously, the roots and the shoot system tend to be pretty well related. Now, one of the things every root has or needs to have is hairs on their roots, especially at the beginning, because that allows for more absorbing capacity. Basically, you end up with more surface area to get that water/nutrient that you need. If you don't have those root hairs, then it's not going to work. Now, if you've got a really big tree, the root hairs are there, they just tend to be bigger themselves and have more smaller offshoots as well. Okay, and so there's different types of roots. So you see the taproot right here. So if you've ever eaten a carrot, that's part of a root system. And that's a very classic case of a taproot. And then you've got these other ones. This is a prop root as well. So on the right side here, prop roots can be used for the plant to kind of help stay stabilized. And then you can have specialized roots like in black mangroves, which are aquatic trees that live like at the island. When you're driving into the island to the right, there tends to be all these like bushes growing in the water. Their roots have these sections called nematophores, where they stick up out of the ground to help the roots absorb oxygen in a really salty environment. Okay, and then of course perennial plants, they regrow each year from the root coming in and out. So perennial means that it's growing all the time. Annual means that it's growing once a season. So that's the differences there. And so there's different zones to know. So obviously the root cap is a big one. That's the part that's protecting the main meristem of the root. So where the root's actively dividing and growing with mitosis, that's the root cap. The zone of elongation is what it sounds like. The cells get longer so they can specialize. And then the zone of maturation is where the cells become differentiated. That tends to be more close to the stem, and that's where the rest of the plant matured from. So if you look at it here, in this diagram, you've got the root cap in the bottom. That's the one where you've got the mitosis happening. Okay. Zone of cell division, where it's going to be growing. Elongations, where they get bigger. And then the maturation zone right here, that's where things get really interesting, because that becomes the rest of the stuff you need. And again, in every root, especially this is a eudicot, in every root here, you've got that star-shaped xylem-phloem complex, right? The phloem's that outer blue, and the xylem's that inner red, and that ends up being kind of their trademark here. So here's kind of a closer look, and you can actually see the micrograph of that vascular tissue. And there's that star, right? You've got the xylem in the center with pockets of phloem on the outsides here and here, right? And so you've got that endodermis, which tends to be that middle section, and then the pericycle is the one directly surrounding the vascular bundles and the vascular tissue. And then there's our happy starfish, yay! So there's our eudicot, right? So that's the end of the root tip. Out here this is the protective portion, right? The root cap. But then in here, all these cells are in some phase of mitosis. And so they're able to grow and allow the roots to extend. And that's the zone of cell division. Now in the monocot, they do have a lot of the same tissues, but it's not organized the same way. So in the dicot, you'll remember that there was kind of that pith in the center, right? And then everything else was around it. Well, that's not how it works in the monocot. In the monocot, they've got vascular bundles spread out everywhere, so the pith is just everywhere as well. So if you look at it here, there's the epidermis, the cortex, okay, the endodermis, the middle, and you've got the pericycle, and then right around there, this is the pith, kind of towards the center here, and you've got your xylem and your phloem. So in the dicot, you didn't see much of it because of that star of xylem and phloem in the center, so the pith is kind of a little more clearly there, and it's spread out everywhere. It's not really localized in one region or another. And then again, the tissues tend to be pretty similar in all the root systems. So you've got vascular tissue, which is your xylem and phloem. Those are the bundles. The endodermis is that layer inside, and it tends to be pretty impermeable, and it gets the water and minerals up and through. Okay. Then you've got the pericycle, which is that first layer of cells. That basically starts to allow the branching and the lateral roots to form. The cortex is made out of parenchyma, as we mentioned earlier. Okay, that's that body type tissue, and it tends to also be for storage. Then you've got the epidermis, which is the outer part. It's got the root hairs and the zone of maturation. Now, for plant nutrition, plants need to have certain things. So there's a nice little acronym down there, right? C. Hopkins Cafe, managed by mine cousin Clyde Moe. These are all the different nutrients. So the macronutrients is a fancy term for they need these in large amounts. Plants need carbon, hydrogen, oxygen, phosphorus, K is potassium, N is nitrogen, S is sulfur, then calcium, iron, and magnesium. The micro ones, and magnesium and iron are kind of like a middle ground here, but these micro ones, they need to have it in smaller amounts. You've got boron, manganese, copper, zinc, chlorine, and molybdenum, I believe that one is, right? So these are your nutrients that every plant needs. It just depends on what the plant is, where it lives. It's going to be differentiating the amounts for everything, but probably the biggest ones tend to be C and O because carbon dioxide. And then throw in some of that hydrogen, so those are the main ones. And again, here's nitrogen. This is what happens when your plant does not have nitrogen. It will not grow very well because it doesn't have the nutrients to build the tissue. Okay, nitrogen is one of those protein building tissues along with phosphorus. If you don't have it, then you can't grow. So right here, this is where the solution had the nitrogen. And if you can see how much bigger the plant got because of that. Okay, so right here you can see this is the big adaptation. N2, if you remember a little bit of chemistry, is not a very usable molecule. It's two nitrogens, okay, sitting right next to each other. These are supposed to be electrons. Sorry for the giant dot there. But these two tend to be very unusable. Nothing can use it because of that triple bond. So plants can't, so they end up having to have a mutualistic relationship with bacteria. And the bacteria, what they do, they fix the nitrogen. They turn it into NO3-, that's nitrate. Or I would actually venture to say nitrite is also another important one. That's NO2 -1 as well. And then ammonium is kind of your weaker one too. But these tend to live in these root nodes where there's going to be quite a bit of bacteria to fix that. If you don't, and you're an organism just trying to fix N2, that's the really not a great way to get along. The other adaptation that's really important is the one with fungus. Okay, so fungus help the plant increase water and break down nutrients. So the bacteria was all about getting nitrogen. This is going to be about getting those smaller nutrients that you want. Okay, so the fungi are going to provide them with sugars, amino acids, everything to help them build up. So again, in the presence of this stuff, plants grow really big. In the absence of it, they still grow, but just a lot smaller. And so again, we've been mentioning this several times, so this one shouldn't take too long here, but xylem and phloem. Okay, we've got vessels and tracheids for our xylem, and the main process we're using here is something called osmosis. Right, you're allowing water to go to create this root pressure that's going to allow water to pass through the cells. Okay, and one of the big models you have to understand, this is a big one to make sure you remember, is the cohesion-tension model. The idea is that water gets from the leaf and it creates a tension to make a water column, and that allows for the water to move through. Okay, because as I mentioned earlier, about 90% of the water that gets taken up by the roots goes to the leaves and gets lost. So that lack of water allows at the top allows the water from the bottom to flow up. And there's two different properties of water that are super important here. That's cohesion, where water sticks together, and adhesion, where water sticks to something else. So, if you squirt a water gun at a wall, the droplets don't hit the wall and bounce off, they stick. That's adhesion, right? Cohesion is the fact that water will stick together. If you ever did that chemistry experiment or that middle school experiment where you put drops of water on a penny, those drops of water stuck together because of cohesion. So here's the model, right? Water's lost at the top, and because of that, it creates a space in the water column, and the properties of water allow that to make it fill and go straight up. Okay, and so that's the big deal here, and the idea is the cohesion-tension model. So lack of water up here, a bunch of water lost, lets the water go straight up, and it carries with it some of that nutrient as well. And so how does this happen? How does the water get lost? Well, it's through the opening and closing of stomata. Plants need to be able to do that to get gas in and out, right? They want to be able to get your CO2 and need to get rid of their O2, so they open up what's called their guard cells. That's these things over here. Okay, so the guard cells can open or close depending on the pressure of water. That's called turgor pressure. Okay, so water enters in, and these things stay really kind of open because of that water. Then, as it's open, the water begins to evaporate, and what happens? There's no more pressure. Then the cell itself begins to close off. So at that point, it's now storing the water, and the water it's getting from the roots start to come back up. If the pressure builds again, then the stomata will open. But it's all about that thing called turgor pressure. Now, when we're talking about sugar, the main part that's in phloem, right? That starts at the leaves and goes to the bottom of the plant. Now, that also can happen in the flowers as well, but sometimes the food that's in the roots can go up to the rest of the plant. If it's an immature plant, or if the plant is having issues where it's needing some of that energy, so it ends up going the opposite way. But mainly it's from the top to the bottom, okay? And that's where we use that plasmodesmata. Right? We mentioned that in the previous chapters, but it allows for the cytoplasm to kind of push through these sieve tubes. We saw that earlier in the PowerPoint. So that's the pressure flow model. Okay, so that's a little bit different from the other. Pressure flow model in this case is where the molecules are going to go from a high concentration gradient, the source, to a low concentration gradient, which we call the sink. Not the sink like the kitchen sink, but basically the area for storage. So leaves tend to be where it happens, right? Sugar is transported through there, and the water follows with it through osmosis. And so it creates a positive pressure, and that's what causes the flow to happen. That water builds up, and it builds up too much pressure, so the sugars and follow that water down to the rest of the plant. Okay, and the rest of it is what we call a sugar sink. So it's basically an area for storage. So along the way from the leaf, if their stems are needing energy, it gets stored, a lot of it in the parenchyma. And then that phloem basically continues letting the nutrient flow from source to sink. So here's kind of the picture of it all, so you can see it. So if you have a source, right, that's the top up here, it's going to allow that sieve tube to pass through, okay? And when you hit right here, these spaces in between, some portions of the plant will need it, and there's your companion cells off to the side, remember those? Okay. Some plants will keep that sugar that's needed, and the water will continue to flow. Others may not, and they'll just let it pass through for the next few cells. So, and this is a constant thing. The plant needs to be photosynthesizing all the time. And as you can see, it continues to just flow downwards, and towards the end you just got more water molecules and sugar as well. But as you can see, right, sometimes the water stays depending on what the plant needs, or the sugar will stay depending on what the plant needs. So it just depends on its needs at the time and how much it's able to make energy from the sun. So that's why a plant needs water and sugar at all times. Most of the time this is how they transport it down. And then finally, when you get down to the roots, you've got still some more of these sieve plates, and the sugars end up passing through to the sides as well as some of the water. So water is still important, but again, you got to remember, water is not the source. This is the source of sugar we're talking about. So the sugars now here are getting to the roots, and then the roots themselves are now have nutrients to get to grow and continue living on. And so again, sugar's actively transported down through the sieve tubes through osmosis. So it needs to have something to carry. It's not just like a tube of sugar. You can't just cut the tree and there's like a river of sugar coming out. It needs to have water with it. Then that positive pressure causes the flow to go down, and then finally the sink is stuck, or not stuck, but sent down to wherever it needs to be, and that is still being used through osmosis. Okay, and so hopefully you got as much as you could out of that chapter, and it was helpful. Thanks.